Thank you, Maya. Thank you, organizers, for the opportunity to take part in CASP. Introduction to my team. There's 4 of us. Rachel Kresh, she'll be leading an RNA SIG later today. She's here in person. Philip Pham and Ramya Rangan are also joined our assessment team this year. All 4 of us, as Maya mentioned, are predictors. So we've been very active participants in the RNA puzzles experiments. And also several of us are involved in developing experimental methods for RNA, like RNA cryo-EM. This is the first time that we've been invited to be assessors, so thank you very much for that opportunity. This really does feel like a historic event for the RNA field. Going into CASP, we had 2 big hopes. Okay, one was in terms of pushing forward the methods. So RNA deep learning is very much in its infancy. Some of the work that we did with Ron Dror did show that scoring based on deep learning methods could help pick out better structures. These are the best submitted models for 2 RNA puzzles that happened in 2019 and 2020. And in some cases, the deep learning score helped us pick out really pretty remarkably accurate structures, like shown on the left. But you can also see here on the right, this is the best structure predicted in the field, but it leaves something to be desired in terms of the comparison of the experimental structure and the model, which are shown in green and blue. So we were hoping that with CASP would excite people who had been developing deep learning methods in the protein world to come and develop RNA-based deep learning methods, and we started to see that happen over the last few months. In addition, we were hoping that CASP could be the premier of RNA structure assessment based on cryo-EM. So cryo-EM had not been really applied to RNA extensively before about 2019 or 2020, but it turns out that our cryo-EM is a perfect technique for solving RNA. So for example, here's a paper where we published 11 structures of 11 different RNAs all in a single paper. And it turns out that RNA, for some reason we don't fully understand, has high contrast and is a perfect molecule for cryo-EM. So RNA is setting records for the smallest molecules that can be solved through cryo-EM. And in RNA puzzles, there were no previous targets that were based in cryo-EM, but we thought CASP would be a great forum for those kinds of targets because the work of Maya Toff and others in prior CASPs, at CASP there's a sense for how to handle and assess based on these often somewhat medium to low resolution cryo-EM maps. Okay, so I'm going to give you an overview here. So I think Eric has already sort of given you a beautiful preview. We independently carried out an assessment based on more CASP-like tools. So just one slide overview of everything that happened. In CASP, there was a big jump in the number of targets that could be looked at per season. So typically in a 3-month period for RNA puzzles, you might see, let's say at most, 2 or 3 targets. In CASP, there were 12 RNA-containing targets. And typically in RNA puzzles, there would be on the order of a dozen predictors per target, whereas in CASP, it elicited predictions from at least 40 different modeling groups. And overall, of these 12 targets, the model quality was quite excellent. These are 4 RNAs that were solved by X-ray crystallography. They have atomically ordered structures. And in green is the experimental structure, and in blue is one of the models that was submitted by predictors. Visually, really excellent accuracy. These were solved by X-ray, but the remaining, actually, the majority of the targets were solved by cryo-EM. So here's 6 structures composed of RNA only. Eric's already mentioned these. 2 of these come from viruses, actually 2 coronavirus RNA domains, and 4 of these are synthetic designed RNA nanostructures. And again, visually, there's quite good recovery or prediction of the folds, which are shown in green, experimental structures, and blue is a model that came out of CASP. The last 2 of these 12 targets are RNA-protein targets, and Eric's already mentioned these did not go so well, and I'll come back to this later in the talk. Okay, so we reasoned that we would learn a lot more if we could carry out quantitative assessments to help us bring out themes in the predictions, as has been done in previous CASPs. And so we decided to develop a CASP-style quantitative ranking of RNA models. The one thing we were excited to try was to was to pilot GDT for RNA. So GDT, remarkably has not been applied to RNA before. It's been proposed. But the way that we helped validate the use of GDT was we actually took several of these targets, and then the 4 folks in our team independently ranked them visually compared to the experimental structure. And then we checked what GDT produces, and basically all these cases, the GDT ranking agreed quite well with our human rankings, at least within the uncertainties or the differences between the 4 of us in ranking the models. And so the final score that we ended up with actually does contain GDT. It also contains a few other terms. So like a lot of the other CASP-based metrics, we are using a linear combination of z-scores. We have a few components here. GDT is in the component that tries to capture the global topology. How accurate are the models globally compared to the experimental structure? We also have these, we also piloted LDDT for RNA, which again had not been really used before for assessing RNA structures. And that's to capture local structural accuracy. And we also included a third term here, which is the mold probability clash score, which actually has been validated and used widely for RNA. These 3 terms, as I mentioned, are very much inspired by what's happened in CASP before. We did want to make sure that we complemented the 2 new terms, it's new for RNA terms, with terms that had been developed for RNA before. So we went ahead and included with the GDT TM score, which Yang Zhan's group has calibrated for RNA. And in terms of the local structural score, we decided to include the INF score that Eric mentioned in his talk, which assesses the accuracy of base stacks and Watson-Crick-Franklin base pairs and non-Watson-Crick-Franklin base pairs. Okay, so this is the metric. And here are the, and there's one more piece of the assessment I need to tell you about. So as has already been mentioned, RNA is flexible. And actually for several of our targets, we knew from the experiments that the RNAs could form multiple states. So here's one example for one of these RNA nanostructures. Ebby Anderson's group actually let us know, and we told predictors that there's at least 2 structures that could be captured of the RNA. One of them is a kind of kinetic trap, an early folding intermediate of the RNA, and the other one is the mature state. And we told predictors that they could potentially model both if they'd like within their 5 models. And this is one of these bat coronavirus RNA domains. And here, automated cryo-EM classifications pulls out actually at least 4 different maps that compose that are needed to define the structure. And in this case, we fitted 10 models to each of these maps. So this came from my group working with Wachu. And so we actually had 40 models here as potential experimental structures. So what do we do in this case? Well, we followed a protocol that was very similar to what's happened in prior CASPs, as well in RNA puzzles when there's multiple experimental structures. We did an all versus all comparison of the 5 predictor models to all available experimental models. Then we rewarded the predictor based on their best score from these comparisons. Okay, so here are the overall rankings from this, we call the zRNA metric. And it was pretty clear cut who the best predictors were according to these quantitative metrics. 232, there's a sizable gap to the second best predictor. There's another gap then to the next 2 predictors. There's another pretty big gap to the next 3 predictors. And then there's the rest of them going all the way down, okay? When we first prepared this bar graph, we didn't know who the predictors were. It was, it's a double-blind experiment. But we did want to see whether there were predictors that were doing particularly well on particular kinds of targets. And what we ended up finding is that for these top 4 predictors, and especially the top 3, they basically did pretty well on all the targets. So this is a heat map of the z-scores. Unlike some of the prior talks, we chose to use blue to mean a good, higher than average accuracy model. But you can see that for the targets where these top 4 predictors made a prediction, this predictor didn't make predictions in all the targets, they were basically blue. So they did fine all the way through. We assumed, based on what had happened in previous CASPs, that this is a signal that these groups had come up with very similar methods, which we assumed was deep learning based on the preprints that have been coming out over the last year. But then when we learned who the groups were, we were surprised to see that these top 4 predictors were folks who had published previously, in some cases over 15 or 20 years in the RNA field. And they're going to be our speakers today. Peng Xiang represented the AI-Chemie RNA2 method. Shiji Chen's group at Missouri will be speaking today. RNA Paulus is RNA composer-based methods led by Martha Shashniak's group, and then Gene Silico is Janusz Buniecki's predictions. And actually, some of these have already been mentioned in Eric's talk as prior RNA puzzle predictors. So maybe they've used deep learning, but at least when we were reading the abstracts, we didn't see any mention of it. In fact, out of the 27 abstracts that were described these RNA prediction methods, 18 of them mentioned deep learning. They're highlighted here in orange. So the majority of them have used some sort of deep learning in the scoring or structure generation. But there's a remarkable negative selection in CASP, if you look at the top, where there's no mention of deep learning from the top 4 groups. Okay, so we don't quite know what's happening, which is also why Eric said, this is to be discussed, the influence of deep learning for RNA. One thing that I did want to note was that it's interesting, there's a block of targets here where these top 3 or 4 groups did quite well, certainly better than the rest. And these happen to be these 4 synthetic designed RNA nanostructures where there's no multiple sequence alignment. Okay, so it may be that, speculation that artificial neural networks, which are still relying on MSAs to make initial headway onto an RNA problem. And here, what was required was basically a biological neural network, a human being, to recognize that, hey, this is a nanostructure designed by another human being, and boy, it should have these symmetries and things that I would design into an RNA nanostructure. Okay, speculation. Okay, so let's take a closer look. We want to double check our rankings because this kind of z-score hadn't been applied to RNA before, and some of these metrics like GDT hadn't either. So I'm showing you here the groups again, ranked from 1, 2, 3 all the way down, and we check to see whether if we used other scores for the global topology, do we get the same ranking? And the answer is yes. If you just use TM score, even if you just use good old-fashioned RMSD, okay, you get the same ranking certainly of the top 4 groups with some shuffling below that. What if you don't use global scores, but you use these more local structural accuracy scores, like IN for LDDT? So here, at least the top 3 are always the same as well. So it's really the same ranking. If you look at clash score by itself, it turns out that, that the top few scores weren't particularly great, but they also weren't particularly bad. Okay. And if you could drop clash score and you'd have basically the same rankings. And then last, and Eric mentioned this, we were worried that for the cryo-EM targets, that there might be model bias in the cryo-EM experimental structures. Okay, the methods that are used to build RNAs into these low-resolution maps actually come from packages like Rosetta or SimRNA, and that they may introduce bias into the experimental models. So Rachel Kresh, with a lot of advice from Maya Toff and Tom Mulvaney and Maya's group, has developed a pure map to model analysis that avoids model bias. And there, she'll talk about that more later in tonight. But this again ranks the top 4 groups within the top 4 with some slight shuffling in the order. Okay, so this is just pure cryo-EM and just the cryo-EM targets. Okay, so we think this ranking is pretty robust, despite there being only 12 targets. I'm going to now talk to you about some highlights here, and now I'm going to include a little bit of extra prior information that came from our personal experience with some of these targets or the experience of some of the experimentalists. Okay, so this is the first RNA target. It's this CPB3 ribozyme. It's actually the first RNA-only ribosome and only RNA-only ribosome discovered in humans. It's pretty cool that the crystal structure is the first RNA target in CASP also. What I'm showing you here is in pink were a set of models for this ribozyme that actually my group had on disk because we were modeling this in 2019. And this is using Rosetta fragment assembly based methods. When the crystal structure came out, we were pleased to see that the overall fold was the same as what we'd predicted. This is actually a homology model. But the global fold, this helix is sticking out in the wrong direction. So we assumed that this is some sort of could be explained as some sort of crystallization artifact. Okay, this ribosome crystallizes a dimer, and well, that kind of distorts the overall fold. But look at this prediction from AICME RNA2. It really gets everything right. Okay, and that's probably not a crystallization artifact, the global fold of the RNA. Okay, I'll show you another example here where there's a prior expectation for the structure. This is one of these RNA nanostructures. So I contacted Ebby and asked him, what did you think the structure was going to be when you designed it? So he sent me his PDB file for this 6 helix bundle, and it's got this very symmetric, 6 helix structure. Their cryo-EM structure actually opens up the barrel, and one of these helical struts is at a diagonal. Okay, and And so I think they were a little bit unhappy when they saw this distortion, but it's really kind of amazing that AI RNA2 in their models, including this model 4, it recovers this opening of the structure and this diagonalization of the helix. And you can quantitatively compare these to the cryo-EM structure. And here, the prediction from a CASP group was a lot better than the design structure. The problem isn't totally solved yet. So none of the structures submitted in CASP were closer to another resolved cryo-EM structure of this RNA and early folding intermediate. So you'd really believe that this problem is solved when groups could predict both of these structures. Okay, and I should say this kind of theme of the CASP predictions being better than the original designs was true for the other 3 nanostructures as well. I just don't have time to show you those examples. Okay, and then here's a third of 3 highlights. This is a SARS-CoV-2 domain, RNA domain. Here again, our group had some prior expectation for what this RNA should look like, and we had published a set of structural analyses of different domains of the SARS-CoV-2 genome back in 2020 and 2021. And we had expected for the so-called SL5 domain that it wouldn't have a well-defined structure. Our Rosetta modeling didn't converge. And that's often a signature that an RNA doesn't, well, we assumed a signature that the RNA doesn't have a fixed structure. If it did, it would have one of these sort of H-like folds. That was our prediction. So we were shocked when we then did cryo-EM on this domain and, what did I just do? And it actually has a really nicely resolved map shown here in gray. And there are major grooves and minor grooves, and this is really resolvable by cryo-EM. It has a T-like shape and not an H-like shape. And then some models, including this one from Gene Silico, actually, again, blindly were able to predict that T-like shape, which matches the density well. Okay, and there's a similar story here for a bat coronavirus SL5 domain that we also contributed as experimentalists to CASP. So where are problem areas? Well, as Eric already mentioned, RNA-protein complexes are difficult. So this is an RNA sponge that soaks up protein dimers in bacteria. This is the so-called CSRA system. This is the cryo-EM structure. Xiaoming Su kindly made this available to CASP. This was the best structure based on the RNA TM score that was submitted. It came from the RNA Polis group, and Martha will be talking in a little bit. And you can see it doesn't really have the overall global fold. And we're very curious to see if Martha made any attempt to include proteins. The prediction from the group did not include protein dimers in their CASP models. There were other predictions that did include the proteins in the model, and in many cases they appear to have some details of how the protein interacts with RNA correct, but the proteins aren't bind to the right spots in the RNA. And so again, the overall fold here is way off, as well as the specific interfaces that are being made. Okay, and there are similar problems with an analogous target, R1189, which is actually the same RNA but bound to 3 protein dimers as opposed to 2 protein dimers. Okay, so I'm going to end here with asking a question of how, in an absolute sense, did CASP15 RNA modelers do? So historically, in proteins, we often use for monomer structure prediction, we often use, GDT. Is it above 50, or is it above 85, or is it above 90? The cutoff keeps getting higher and higher. How well did RNA modelers do on that kind of a scale? So we assessed whether folks got TM scores greater than 0.45. This is a threshold that has been defined by Yang Zhang's group for being, at the threshold of close structural similarity, close enough to being a matching template for RNA. For protein, it's 0.5. For RNA, it's 0.45 in their version of the TM score. We also were curious to see whether GDT could be where it was above 50, which is often taken to mean correct topology for proteins. So here's a result. This is the TM score for these 12 different targets. And here's this cutoff for having the topology roughly right for TM score. And you can see that in most of the targets, actually, there were predictions, often by at least one or sometimes several of these top 4 groups that is above this bar and getting the topology right. Okay, the 2 problem cases are here at the end, and these are the RNA protein targets. Okay, so there's still work to be done. And actually, this target is a pre-Q-sine ribose, which is small 30-mer, where the models don't quite meet the line. And this appears to be due to something funny about TM score. I think it's a bit too stringent for small RNAs. If you instead look at GDT, My presentation is stuck. Oh, there it is. Okay, thank you. It turns out that GDT shows that for that RNA case that there's plenty of models that are above GDT of 80 actually. So these are probably fine, and this is just an artifact of the TM score. So overall, in terms of just global topology wise, all of the submissions for RNA only targets, all 10 of them, there are submissions that have TM score greater than 0.45 or at least GDT TS of greater than 50. That's pretty good. Then there's a question is for RNA, like how good could you get? Like, we have a rule of thumb for proteins that GDT greater than, I don't know, 90, 95 is as good as you can get, given, if you have proteins captured in different crystals, for example, they're different by that amount of GTT. So what we did was for some of these cases, we have multiple alternative conformations solved by cryo-EM or an RNA in 2 different copies in 2 different asymmetric units. So we use that to estimate how good could you get. And in most of the cases, actually, the modelers haven't reached that threshold for experimental uncertainty, getting within experimental uncertainty. There actually are a couple of cases where predictions go above this bar, but these are actually the most flexible RNAs. They're the ones solved by cryo-EM, where there's like very clearly very different looking folds captured by cryo-EM. But I think overall, we're not nearly at the level of where proteins is, where you just, you're within experimental uncertainty. You don't, in some ways, have to do the experiments anymore. Okay, so here's a summary. There were 12 really interesting targets in CASP for RNA structure prediction. Most of them came from cryo-EM. And there were 40 predictors, and most of them, at least to us, appear new to the RNA field, and that's tremendously exciting. It turns out the class of CASP metrics are useful for RNA assessment, even for some of the more flexible targets. And I want to highlight that GDTTS and LGC are actually, like, they're pretty useful. Potentially in future CASPs, they could be used universally for example, if we're looking at protein RNA complexes or perhaps protein and RNA alone, all together in one category. The top 4 predictors from the zRNA analysis, they're pretty unambiguous, even if you change out the metrics that you're using. And we're going to hear from all 4 of these later today. For the 10 RNA-only targets, there was at least 1 good model for each target. In several cases where there was a template or there was a design structure, there's clear improvement over those prior structures. And in all of these cases, there's at least 1 model with a TM score greater than 0.45 and or a GDT greater than 50. At the same time, they're not quite at the level of accuracy where you wouldn't have to do the experiment. The similarity to the closest experimental structure of a prediction is still typically worse than the similarity between alternative experimental structures when we have those. RNA protein complexes remain challenging. I put an asterisk here because I want to say there's one category of RNA that actually we didn't have in CASP. And Eric alluded to this. So there are RNAs that, for example, bind ligands and they form really beautiful atomically ordered structures that, and it's the same structure when different groups solve the structure in different crystal forms or from different homologs. They're really well defined, and we didn't have any of those here in CASP, or we had them, but there were templates available. So I think for those hard targets, we're going to have to, the jury is still out. We're going to have to wait for the assessments of ongoing RNA puzzles or for the next CASP. Okay, so I'm going to end here by thanking all the people who've helped us, Eric and Chichao, Martha, Maciej, Maya, Tom, Gabriel, Martin Magnus, Adam Zemla, Cheng Xinjiang. They helped us develop these software packages for doing the assessments. Nick and Lisa gave us advice on how to do a CASP-like assessment this year, and of course, Andrei, Christoph, and John for the invitation to assess. This is us. This is our funding. And also, if you're interested in pushing forward prediction or cryo-EM of RNA, I invite you to email us. We're recruiting folks to our group. Thanks.
Thanks a lot, Rhiju. Questions, please.
So outside of the designed RNA, which I guess likely have pretty regular secondary structures, can you comment?
Who's talking?
So, hi.
Oh, sorry. Thanks.
Can you comment on, I guess, the impact of how or the correct secondary structure assignments for the quality of the downstream models? Do you think it's a matter of just using your, thermodynamic model and, using some automated prediction? Or do you think maybe it's more critical for like some chemical probing derived like more precise secondary structure assignments to get a higher quality model.
Yeah, I didn't have the summary graph here, but for those in basically all 10 RNA only structures, the top groups were able to just nail the secondary structure.
So
And whether that's through covariance or more likely reading papers or just, I don't know, intuiting what the designers were trying to build, that worked. I don't actually think, and based on my own experience as a predictor of RNA, just using a thermodynamic automated secondary structure predictor is typically not good enough to get the right answer for most of these targets. So I think there's this critical piece of human intuition there that's also does not appear to be fully captured in more newer neural network approaches for RNA secondary structure. There's artificial neural network approaches. So at least currently, I mean, that's a big opportunity actually, because you still need a human neural network to go and figure out the secondary structures.
So was there some literature available regarding some of these targets?
I'm sorry, ask a question.
So was there some, literature available for these targets, like some shape data or whatever?
Yeah, for the natural ones, you could typically find there was literature. In some cases, there was chemical mapping data already available that could help you hone your secondary structure. In some cases, there were so-called RFAM families where you could look at an alignment also as well. For the synthetic ones, there wasn't that any of that literature. But again, I think a human being who is familiar with RNA or RNA nanostructures would be able to guess the secondary structure.
Any other questions? Oh, John.
So of course, it's nice that you use GDTTS, but there seemed to be quite a big difference in the summary plots you showed between the distributions for TM and GDTTS. Do they give the same picture, or are they really?
They give the same rankings, at least in the top. And then, let's see here.
Well, I was thinking of you had the two panels with the summary scores for each target.
Here's GDT, here's TM. So there's a switch in third and fourth, and then there's some shuffling of the next three.
Yeah, I was thinking more of between individual targets.
Oh.
That when you switch from TM.
Oh, yeah. Yeah.
Up and down somehow.
Yeah, I think.
Like
I'm going to talk more about this later.
And some didn't. Right?
In the Ensemble wrap-up session. I don't think that either of these scores is really the right thing to be doing for some of the more flexible RNA structures. I don't think RMSD is either. So I'm not sure if there really is a single score. I think that direct comparison to the data is the way to handle the rankings, and there's a clean way to do that for cryo-EM at least. Yeah, I wish there was just like a score and you could say, well, once you get above 95, then you're done. But I don't think there is one. Yeah, LDGT may be a little bit better, but I don't know.
No, I wasn't trying to push it. It's better, but just 1116 and 1117 sort of swap when you change metrics, right?
Yeah, I think this again, like I'm saying, this is a sort of a bug, or I don't know, maybe a feature in the TM score. It's very stringent for very small RNAs to decide what's close.
One last question.
Very short and very simple question. So for proteins, we have culprits, when the target is difficult, right? MSA, homology. So is the situation pretty much the same for RNA? What makes an RNA target hard target?
Yeah, I think presence of structural templates and MSAs, they can give you some, but some of the most remarkable predictions here happened for these synthetic nanostructures where there's no MSA. So it's and I think those are just these funny cases where a human being can go in and grok what the designer wanted to make. And then so I think that there's no single way to define that we could figure out to define difficulty.